Hello, everyone, and welcome to today's live broadcast, RNA Targeting CRISPR and Application to Diseases, presented by Dr. Jean Yeo, Principal Investigator, Professor, UCSD. We are excited to bring you this educational webcast presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. I'm Julie Simmeroth of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Please join me in welcoming Dr. Jean Yeo. I will now turn the presentation over to him.
Thank you, Tiffany, for the introduction. So I'm happy to present our work from my lab. All right, so the title of my talk is RNA Targeting by CRISPR-Cas. And I will go over some of the projects that we are using this application for technology for. Okay, so if I just give you a rough idea of what my lab is doing, here's some background. My lab is interested in studying RNA processing, and RNA processing, as you can see here, is the process by which RNAs are modified and matured after being transcribed from the genome. And for the most part, RNA is then transported to the cytoplasm and translated into protein. Now, RNA processing is diverse, and there are many aspects of RNA processing, ranging from splicing to polydenylation, small RNA processing and biology, RNA transport, decay, RNA editing, and assemblies of protein RNA complexes. And so my lab focuses on the discovery of mechanisms that underlie RNA processing, as well as the regulatory networks that are controlled by proteins, RNA binding proteins that bind and process RNA. The other part of my lab worked on strategies to manipulate RNA processing using a variety of different technologies. And through the last decade of the work in my lab in this field, we have been applying and developing novel computational, molecular, and cellular tools to aid our process in understanding RNA processing. So just to move ahead, here's some background on our recent work in our efforts in large-scale analyses of RNA processing. So RNA processing defects underlie a lot of genetic and somatic diseases in humans. And here are just some examples, not comprehensive, but a good repertoire of diseases that are caused by altered RNA biology. Now, again, just as a reminder, we study how RNA is processed, and the biggest class of proteins that control RNA processing are called RNA binding proteins. There are many RNA binding proteins are encoded in the human genome, and they can bind and directly to RNA to control how RNA is modulated. Here is a rather complicated figure, but comprehensive one, from Tom Tuchel's lab that shows the many aspects in which RNA can be processed and bound by RNA binding proteins. So just to remind us again, RNA binding proteins play critical roles in development, and mutation or alteration of these RNA binding proteins cause human disease by altering RNA processing pathways. As a reminder also, these RNA binding proteins exert their effects by interacting with short cis-regulatory elements that are encoded in mRNA or pre-messenger RNA. And these short cis-regulatory elements can be sequence-specific recognition elements, or it can be RNA secondary structural motifs that are embedded within the RNA molecule. Now, by binding to these short cis-regulatory elements, RNA binding proteins can mediate a variety of the different RNA processing tasks. However, currently, the functions of the majority of RNA binding proteins are still not understood, and the precise RNA targets which are bound and regulated by these RNA binding proteins are largely unexplored. And from early studies in the field, we expect that the majority of RNA binding proteins can interact with hundreds, if not thousands, of different RNA substrates. So we have been continuing to improve methods to identify the precise binding sites for RNA binding proteins as a way to build a map for what these RNA binding proteins can be doing. We have leveraged technologies such as CLIP, first developed in Bob Darnell's lab in Rockefeller. And CLIP stands for cross-linking IP, immunoprecipitation, where you UV cross-link proteins to RNA and digest away all the unprotected RNA, and then use antibodies to immunoprecipitate the RNA binding protein RNA complexes, and then ligate adapters to specific RNA molecules that are protected by the antibody, by the proteins, and then sequencing the footprint of where the RNA binding proteins bind. So as you can see here on the left, these are polyps of reads that correspond to specific RNA binding protein binding site TDP43. So we have been developing computational methods to analyze large-scale CLIP sequencing data sets, and are also developing new ways to improve these methods to enable large-scale analyses of RNA binding protein function. Now, we know that there are 1,500 human RNA binding proteins, and that's probably a lower estimate. You know, we and others are finding new ones all the time. So there must be better ways to evaluate all these RNA binding proteins more systematically, and also at a pace in which we can then study more biology related to these RNA binding proteins. So over the last year, my lab has published several publications that illustrate our efforts in what we call scalable analyses of RNA binding proteins. For example, on the top left, we have been expanding the list and repertoire of RNA binding proteins by developing algorithms that mine existing protein-protein interaction data sets to identify novel RNA binding proteins conserved in yeast, in fly, and humans. We have also been developing antibody resources for the large-scale study of RNA binding proteins. And here we have manually validated about 700 to 800 antibodies specific for different RNA binding proteins, and identified about 438 antibodies, which are IP grade antibodies for about 400 something RNA binding proteins. And these have now been made all publicly available, where you can search for your favorite antibody that we have already validated using shRNAs and IP followed by Western blotting. On the bottom left, you can see we've also developed enhanced or more robust CLIP methods where we can reduce the number of PCR duplicates. And these are violin plots at the bottom, where each dot corresponds to downloaded data sets for CLIP sequencing representing different RNA binding proteins. And on the y-axis is the usable fraction of reads, reads which are useful and are not simply PCR duplicates. And so you can see on the blue dots on the left that our enhanced clip or eClip method is able to generate data sets with much higher usable read fraction, about 85 and 90% on average, than existing data sets represented by the public iClip and public clip data sets. And finally, we've been developing integrative computational analyses to combine different data types and different technologies to help us make sense of what RNA binding proteins are doing. And just to summarize, over the last 5 years now, we have worked very closely with labs of Brenton Graveley, Chris Birch, Xiangdong Fu, and Eric Lupier to amass a large amount of data sets, about 1,000 replicated data sets that measure different aspects of RNA biology. This data set represents 352 RNA binding proteins, of which 23% of them have no known function. And so we again integrate methods such as RNA-seq, generated by Brenton Graveley's lab, RNA bind and seq, which is a form of CLEX, generated by Chris Birch, ChIP-seq by Xiangdong Fu, and then immunofluorescence data sets generated by Eric Lupier's lab, and our own data sets, eClip data sets for RNA binding proteins that together enable us to understand the localization of RBPs, the RNA dependence of RNA regulation by these RNA binding proteins, and to give us some new ideas about the functions of these RBPs. This is the largest data set of RNA binding proteins systematically collected to date. Everything is publicly available on the ENCODE website, and these data sets generated in these two cell lines, K562 and HepG2. As an example of what the eClip or mapping these RBPs can be useful for, so here one example is eClip data sets for different RNA binding proteins, Lin28B, RBFOX2, PRP8, E2AF2, and IGF2BP1. These different RNA binding proteins bind to diverse places on the same RNA transcript. And the peaks or the high numbers of reads tell you that they bind very specific places, but also places which are unique to the different RNA binding protein. So for example, IGF2BP1 binds to the 3' untranslated region of this gene, whereas RBFOX2, for example, binds to intronic regions flanking exons within the same gene. What can we do with these maps? So we are very encouraged by the fact that last year, Adrian Craner effort at Cold Spring Harbor to study the splicing of this SMN2 gene has enabled Ionis Pharmaceuticals and Biogen to develop antisense oligonucleotide therapies for spinal muscular atrophy. And so in spinal muscular atrophy, loss of function of SMN1 leads to a reduced level of SMN protein, which then leads to spinal muscular atrophy. Now in humans, another copy of the gene SMN1 is present called SMN2. However, SMN2 is alternatively spliced, where one exon is not included in the majority of the SMN2 mature mRNAs. And so it does not produce the correct or sufficient SMN protein. So what Adrian and colleagues have found is that you can develop antisense oligonucleotides that block the binding of an RNA binding protein, HNRPA1, from binding downstream of this particular exon, number 7, I believe, in SMN2, and by blocking A1 from repressing exon usage, you can reverse the splicing to generate 90% of the correct full-length isoform. And this leads to the increase in SMN protein, thereby curing spinal muscular atrophy. We believe that these RNA binding maps that we and others are generating can serve as a potential basis for rational drug design, where we can use the maps to decide where specific RNA therapeutics can be targeted to, such as these antisense oligonucleotides. Okay, shifting gears, I will now move towards talking about the manipulation of RNA processing instead of discovering the maps and where RBPs bind to. So for the past now 5 to 10 years, there have been many studies in the literature using different RNA binding domains to engineer and try to manipulate RNA in living cells. So you can imagine that RNA binding domains within existing RNA binding proteins have evolved to recognize specific cis regulatory elements and sequences. Now one can then construct combinations of these domains to then target any RNA sequence you want. That is the dream. And the best example of this is the puff proteins, where several labs have now assembled different domains in the puff proteins to recognize stretches of 8 or more bases in RNA molecules. Now, if we can engineer RNA binding domains to bind to RNA in specific places, then we can recruit effector domains to manipulate the RNA. This will have tremendous importance and flexibility in being able to understand RNA processing, but also as potential therapeutics. Now, we were very excited when the CRISPR-Cas9, for example, complexes were discovered or were identified. Because if you look at talins and zinc fingers, they're analogous to what the field has been trying to do with puff proteins, which is to assemble proteins to bind to specific nucleic acids. However, the CRISPR-Cas9 technology has now enabled a programmable recognition far more efficient by using a Cas9 protein and a guide RNA to recognize specific DNA sequences. So we envisioned that we could do something similar called programmable RNA recognition by now targeting the CRISPR-Cas or other Cas proteins to RNA instead of DNA. And you can fuse the Cas protein to different effector domains, then you should have the ability to engineer transcriptomes. As a proof of concept that this can work in vivo, we collaborated with Jennifer Doudna's lab, who had shown in 2014 that you can use a PAM oligomer, DNA oligomer, to trick the Cas9 protein to now recognize RNA. And you can have design components of the PAM and PAMer and the guide RNA to now hybridize to specific messenger RNA sequences and thereby targeting the Cas protein to RNA in vitro. Now, in the collaboration with Jennifer, we've decided to see if this also works in vivo in live cells. And as a proof of concept, my student Dave Nellis in the lab fused GFP to a nuclear localized Cas9. And this protein is dead, so it does not have the catalytically active site, which cuts DNA. So we reasoned that if we can design and modifications of the PAMer and the sgRNA to hybridize to express mRNAs in human cells or live cells, then GFP Cas, which is normally nuclear, will follow or will track with the mRNA into the cytoplasm. And here's the proof. As exciting data, we can see on the top left of the boxes here that with a targeting guide RNA and the targeting PAMer that recognizes GAPDH mRNA, the normally nuclear localized Cas9 GFP seen on the bottom right is now moving from the nucleus of the cytoplasm as seen on the top left. And so this is quite exciting because this gives us the first example that the CRISPR-Cas9 system can recognize endogenous mRNA in live cells in a programmable fashion. So this was published in Cell 2016. And in my lab, we decided to then push ahead with the technology to see whether we can identify other RNA sequences and activate different activities in them. So we started looking at microsatellite associated diseases where there are expanded DNA repeats encoded in the human genome, but in patients with different microsatellite diseases such as Fragile X tremor associated syndrome, SCAs, and other diseases, these repeats are expanded to lengths far beyond what we see in normal patients. So in this talk, I'll focus mainly on myotonic dystrophy 1 and myotonic dystrophy 2, so DM1 and DM2. So in DM1, you have these expansions of CTG repeats in the 3' UTR of a gene DMPK. And in DM2, you have a slightly different repeat, CCTG, which is expanded in an intron of a different gene. Now, in both of these diseases, it's been quite well established that these DNA repeats are expressed because they're within genes which are coding genes which are expressed, but they're expressed, and because they're expressed and they're so repetitive and expanded, they sequester different proteins that bind RNA. They sequester these proteins to these repeats, right? So for example, at the bottom here, you can see that muscleblind 1, 2, and 3 are sequestered by these expanded repeats away from their normal targets, where muscleblind normally affects splicing, and thereby you have this disrupted splicing in the cases in cells of patients with myotonic dystrophy. And these repetitive sequences are so expressed, and because they're so repetitive, that you can do RNA fish, which is the image on the left in the nucleus, and see that these repeats are in this RNA foci. Now, we rationalize that we should be able to make an impact in studying myotonic dystrophy, but maybe even helping to reverse some of the RNA toxicity that's derived from these expanded RNA repeats. Now, myotonic dystrophy is a terrible disease. It's a disease of the muscle, but also CNS in the brain. Patients can have cardiac conduction defects and also cataracts, and there are congenital versions of DM as well. So we tried to use rCas9, RNA targeting Cas, fused to GFP again, to target these repeats in cells transfected with just CUG expansions. Without the targeting guide and targeting PAM oligomer, rCas9 localization is diffused. But if we have targeting guide to the repeats and targeting PAMer, we're able to localize Cas9 and GFP to the same places where the RNA foci are, as you can see from the second row, where they're well co-localized. Now, surprisingly, probably because of the repeat expansion, the repetitive nature of the expansion, we do not require the use of the PAM oligomer, and we're still able to co-localize Cas9 to these RNA repeats. Now, the next step, we were inspired by a study by Zhefeng Wang's lab, and we used this PIN endonuclease domain and now fused to DIT Cas9, and we envisioned that this endonuclease would, if brought close to these RNA foci, will lead to the destabilization of these RNA foci. Now, in our study, DCAS9 alone can destabilize RNA foci, but the PIN enables us to achieve this at much lower doses. So at the bottom row here, you can see with the white arrows that cells expressing high levels of DCAS9 with PIN And you can visualize this with an antibody recognizing the HA epitope fused to DCAS9 pin. So in green, high levels of DCAS9 pin. You can see on the second column that in green that in the left column that you have lower levels of correspondingly lower levels of the RNA foci demarcated in red by RNA fish. So this is the indication, first indication that rCas9 can lower RNA foci of these microsatellite repeat sequences. We can also show that this is true for different repeats, such as CCUG for DM2 and GGG-GCC for ALS. We can quantitate this by counting the number of cells which contain fish signal normalized to the repeat only signal. And you can see that for many different repeats, including CAG, which is present in Huntington's, we can lower the number of cells with these repetitive sequences. Now, just to show that these are RNA dependent, we have used a construct which is DOX inducible, where upon treatment of doxycycline, you can use GFP on one side of the construct, and then on the other side, you can derive part of the DMPK gene encoding about 1,000 CTG repeats in the 3' UTR, which is the same, obviously, as DM1. Now, we can then stop doxycycline, and so RNA and then obviously GFP are now transcribed and present in the cells. But after stopping doxycycline, there's no more transcription of these RNA or the GFP, but they are present in the cells already, and we can then transfect in, DCAS9, the pin, and gut RNAs targeting the repeat to show that we can destroy or reduce the foci independent of transcription. And these are just ways of quantitating that number of cells, but also the fold change over DOX, negative DOX. Finally, we can also do this in patient cells, so not just transfected cells, but patients myoblasts from patients with DM1. And on the row at the bottom, you can see that in cells that express DCAS9 pin, HA tag, we can reduce the RNA foci. We believe in vivo at low doses, the pin fusion can degrade RNA much more efficiently than DCAS9 without pin, even though DCAS9 by binding, we believe, can unravel the RNA structure and make available endogenous processes to lower the RNA foci. Now, we also know that muscle-blind protein can co-localize with the repeats in cells from DM1 mice models. And so we wanted to know if we can destroy the repeats, can we redistribute muscle-blind proteins back to their normal localization states. So here showing that muscle-blind normally is in the nucleus with a beautiful, slightly lower cytoplasmic localization pattern. If you express repetitive sequences, muscle-blind is sequestered to the repetitive sequences and co-localized with these RNA fish images, right, on the first column. And if you can treat the cells with rCas9 targeting the repeats, you can now achieve redistribution of muscle-blind back to the normal localization. Now, one would imagine that if you can reverse muscle-blind localization, then you can also reverse alternate splicing patterns, such as present on the left here, where there are different bands showing different isoforms. And you can see in the the DM1 myoblasts from patients, CDG targeting guide RNAs can restore the aberrant splicing patterns from the non-targeting ones back to a normal or controlled wild-type ones. These can be looked at also using large-scale RNA sequencing, and maybe best to summarize globally across all the different isoforms in genes that we can measure. Each dot on the top left represents a % splice in or exon inclusion of of a specific exon in DM1 patients, different from control, and there are lots of dots that are different, right? Lots of exons which are spliced differently. We've removed the ones in the middle, diagonal, which are not different, and in the bottom row, we treat with the CDG targeting guide, and the same dots from above are now moving to the middle, showing that we can reverse transcriptome-wide splicing. This is true both in myoblasts and myotubes, and which is very exciting because these are the downstream molecular markers for that are relevant to the disease. And so this paper was published recently in Cell, to showing that we can use RNA targeting CRISPR-Cas to ameliorate microcellulite repeat expansion diseases. But this is all in Cell, so what about in vivo? So recently, in collaboration with James Thomas and Maury Swanson's lab, we have generated vectors encoding Cas9 pin and guide RNA targeting repeats, and these are packaged into AAV viruses and injected contralaterally in mice models of DM1 expressing the CTG repeats. And we can show that in vivo, we can also reduce the RNA foci in muscles injected with the AAV viruses. These treatment and reduction of the foci also reverse downstream muscle-blind 1 mediated splicing defects, as you can see from the pin and targeting guide treatments. Now, in the first row of the RT-PCR images, that is a reversal splicing back to wild type of the chloride channel gene, which actually restores the correct protein abundance of chloride channel, which is now represented in green by the immunofluorescence images on the right in tissues. We can also show that we can reduce aberrant myonuclei in DM1 mice model tissues, and finally reverse transcriptome-wide splicing defects as well in vivo. So we're very excited because we think that this works in vitro, in patient cells, in vivo, in mice, and there are many other things that we can use RNA targeting Cas for, which I'm happy to elaborate on. But of course, we have to thank the folks in the lab that did this work, right? And so the CLIP and ENCODE related projects have been largely led by Eric Van Nostrand, a talented postdoc in my lab, together with Gabriel Pratt, a graduate student, and supported by bioinformatics staff, as well as a dedicated team of clippers that perform these experiments, Stephen in biology, and so on. For the RNA targeting Cas work, we have Dave and Nellis and Ron Batra, my lab that co-led the work for DM1 and DM2, and together with in collaboration with Kevin Corbett and Maury Swanson. And I'm happy to thank the rest of my group, and thank you for the invitation, and I'm happy to take questions. Thank you very much. And I'll leave my slides here.
Thank you, Dr. Yo, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up with you via email after the presentation. Let's get started. Our first question is, are RNA binding sites preserved on the same mRNA in different tissues?
So that's a good question. That's a good question. So in the ENCODE project, we had mapped RNA binding sites for many RNA binding proteins in 2 cell lines, and evaluating or comparing the RNA binding sites for the same RNA binding protein in both cell lines, we find that many of these sites, the majority of them, are actually preserved. However, when we move to other tissues and probably more relevant tissues and other cellular states, say neurons or embryonic stem cells, we find that there are other RNA binding proteins that are not expressed, obviously, in the lines that we have performed our previous ENCODE experiments, these RNA binding proteins have new sites. There are also RNAs are expressed in a tissue-specific manner, which are now present, which obviously also have new sites. And so I think that a lot of work can still be done for mapping RNA binding proteins in a variety of different systems and tissues.
Thank you. What is next for these large-scale RNA binding protein mapping projects?
So we have continued to now increase the number of reagents, antibodies, and also tag lines for other RNA binding proteins. We want to expand the list of maps for more RNA binding proteins, but also across, as I mentioned, more tissues and cell types. And I think what's also very exciting is to be able to map these RNA binding proteins in different cellular states. So, for example, cells that are treated with different stressors, cells that are models for aging, for example, cells from disease patients with mutations in RNA binding proteins, cells in cancer, cells undergoing chemotherapeutic treatment, for example. And so that all these different states may help us think about how the RNA maps are changing over time, and may give us better indication of which target RNAs can be interrogated for disease manipulation.
Thank you. What are the advantages of using RNA targeting CRISPR versus DNA targeting CRISPR?
So the differences between using RNA targeting CRISPR and DNA targeting CRISPR definitely relies on the application. So DNA targeting, as is currently used for work in the lab or potential in vivo therapy, ex vivo therapeutics, rely on DNA repair, and many post-mitotic cells, neurons, muscle, do not really have efficient DNA repair, and so RNA targeting does not rely on the endogenous DNA repair machinery, and is able to still manipulate and destroy or change RNA processing, which are the downstream of the gene sequence encoded in the genome. The other advantage of RNA targeting is that the off-target rate is presumably quite different. So in DNA targeting, particularly of these microsatellite repeat expansions, there are many of these repeats present across the genome. And one would envision that if DNA cutting happens in many of these repeat sequences in the 3 billion base pairs in the human genome, you will have a lot of aberrant cuts, which may lead to chromosome abnormality and other issues. Whereas the RNA off targets are constrained to the expressed and relevant set of RNA in that particular cell type, and also the on-target sequence that the expression of the abundance of the RNA that we're targeting may be hundreds to 1,000 of copies more than off-target RNA. Whereas in DNA, it's pretty much the same copy number between on and off targets. So there are other advantages as well, but these are the two main ones.
Great, thank you. Next question is, what are the major challenges to using RNA targeting Cas to gene therapy?
The main challenge to using RNA targeting Cas for gene therapy applications has to do with the immunogenicity of the protein. And this is, after all, a bacterial protein, and will be in some cases immunogenic. And so being able to humanize or modify with protein engineering the Cas protein is a opportunity, but also a major obstacle to getting this to work in gene therapy. The other challenge is delivery, but this is true also for other gene therapy applications, where getting the protein into the right cell and having it have a durable response for long periods of time, and getting enough of the protein in as many cells as we need to, that's always going to be, at least for now, a challenge in the gene therapeutic space.
Thank you. We'll wrap up with one last question, and that question is, what are other potential applications to RNA targeting Cas?
So RNA targeting Cas can be used to reduce, as I had shown, the levels of toxic RNA, but it can also be used to change the translation of mRNAs, affect splicing, affect RNA editing, methylation, localization of RNAs. And so it all depends on the effector domains that my lab has a lot of experience with from RNA binding proteins to modify the function of RNAs. And so there are many potential applications for basic research, also therapeutics by modifying RNA. And I think also in the space of reagents and agriculture, where one would want to target or be able to visualize RNA or alter or destroy viruses that affect plants and so on. So I think there are many potential applications to RNA targeting Cas.
I would like to once again thank Dr. Yo for his presentation. Do you have any final comments?
No, thank you for your for the opportunity, and I'm happy to take emails in the future if anyone has other questions. And yeah, thank you for the time.
Thank you once again, Dr. Yo. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 28, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.